This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is the functional organization of a eukaryotic gene? Answer: a eukaryotic gene is composed of several functional regions that control its expression. These include the promoter, which controls the initiation of transcription, the coding region, which contains the information for the synthesis of a specific protein, and the regulatory regions, which control the rate of transcription and the stability of the mRNA. Question: can you explain the role of the promoter in the functional organization of a eukaryotic gene? Answer: the promoter is a DNA sequence located upstream of the coding region of a gene. It serves as a binding site for the RNA polymerase enzyme, which initiates transcription of the gene. The promoter also contains regulatory elements that control the rate of transcription by binding to specific transcription factors. Question: what is the coding region in the functional organization of a eukaryotic gene? Answer: the coding region of a eukaryotic gene contains the information for the synthesis of a specific protein. It is composed of exons, which are transcribed into the mRNA and translated into protein, and introns, which are transcribed into the mRNA but are removed by splicing before translation. Question: can you explain the role of the regulatory regions in the functional organization of a eukaryotic gene? Answer: regulatory regions are non-coding sequences located near the promoter and the coding region of a eukaryotic gene. They control the rate of transcription and the stability of the mRNA. These regions contain cis-acting elements that bind to specific transcription factors, which can activate or repress transcription. They also contain trans-acting elements that bind to RNA binding proteins, which can affect the stability and translation of the mRNA. Question: how does the functional organization of a eukaryotic gene differ from that of a prokaryotic gene? Answer: the functional organization of a eukaryotic gene is more complex than that of a prokaryotic gene. Eukaryotic genes typically contain multiple exons and introns, whereas prokaryotic genes are usually composed of one continuous coding region. Additionally, eukaryotic genes have a greater degree of regulation, with multiple cis and trans-acting elements that control the rate of transcription and the stability of the mRNA. Prokaryotic genes have less complex regulatory regions, which are usually located near the promoter. Question: how does the process of transcription in eukaryotic genes differ from that in prokaryotic genes? Answer: the process of transcription in eukaryotic genes is more complex than in prokaryotic genes. In eukaryotes, transcription is performed by 3 different RNA polymerase enzymes, each responsible for transcribing different types of genes. Additionally, eukaryotic transcription involves the formation of a pre-mRNA molecule, which is then modified by the process of splicing to remove introns and join exons. In prokaryotes, transcription is performed by a single RNA polymerase enzyme, and there is no splicing of the mRNA. Question: can you explain the process of splicing in the functional organization of a eukaryotic gene? Answer: the process of splicing in the functional organization of a eukaryotic gene is the process of removing introns and joining exons from the pre-mRNA molecule. This process is performed by a complex of small nuclear RNAs and proteins called the spliceosome. The spliceosome recognizes specific sequences, called splice sites, at the boundaries of the introns and exons and cuts and rejoins the pre-mRNA accordingly. This results in the formation of a mature mRNA molecule that can be translated into protein. Question: how does alternative splicing affect the functional organization of a eukaryotic gene? Answer: alternative splicing is a process by which different exons or parts of exons are included or excluded from the final mRNA molecule. This results in the production of different protein isoforms from a single gene. This process increases the functional diversity of a eukaryotic gene by allowing for the production of multiple proteins from a single gene. It also provides a mechanism for regulation of gene expression at the level of mRNA processing. Question: how does post-transcriptional regulation affect the functional organization of a eukaryotic gene? Answer: post-transcriptional regulation refers to the mechanisms that control the stability, localization, and translation of the mRNA molecule after it is transcribed. These mechanisms include the addition of a poly-A tail to the 3' end of the mRNA, which increases increases its stability, and the addition of a cap structure to the 5' of the mRNA, which is necessary for translation. Additionally, mRNA can be localized to specific regions of the cell, such as the cytoplasm or the nucleus, which can also affect its stability and translation. These post-transcriptional mechanisms can also be regulated by cis and trans-acting elements, which can affect the overall expression of a eukaryotic gene. Question: how do epigenetic mechanisms affect the functional organization of a eukaryotic gene? Answer: epigenetic mechanisms refer to the heritable changes in gene expression that do not involve changes to the underlying DNA sequence. These mechanisms include DNA methylation, histone modification, and chromatin remodeling, which can affect the accessibility of the DNA to the transcriptional machinery. Epigenetic modifications can either activate or repress gene expression and can be passed on to the next generation of cells. This means that the functional organization of a eukaryotic gene can be affected not only by the DNA sequence, but also by the epigenetic modifications that have been made to it. Question: how do non-coding RNAs affect the functional organization of a eukaryotic gene? Answer: non-coding RNAs are RNA molecules that do not encode proteins. These include microRNAs, small interfering RNAs, and long non-coding RNAs. ncRNAs can regulate gene expression at different levels, by binding to the mRNA and affecting its stability, translation, or by binding to the DNA and affecting its accessibility to the transcriptional machinery. This means that ncRNAs can affect the functional organization of a eukaryotic gene by modulating its expression and influencing the production of its protein product.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the First Aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.